This is a production of Cornell University.
Okay, cool. So I like to walk around a little bit. Hopefully that's okay. So plant roots. I assume most of you in this room know what plant roots are, right? But what you may not know is that there are different types of plant roots, and these different types of plant roots have different functional roles in the environment. And we think that these different functional roots, or different roots with different functionalities, lead to different associations of microorganisms. And today I'm going to be talking about two separate projects where we've generated some evidence to show that appears to be the case. So plant roots, I probably don't have to convince anyone in this room that plant roots are incredibly important for a number of different ecosystem services, including nutrient cycling and carbon cycling. In particular, the fine roots of a plant, they disproportionately impact carbon cycling despite their very tiny size. And we also see roots as, being as really important for soil stability. We see them as a little bit as ecosystem engineers. As the plant root is growing through the soil profile, they're able to create pore space and weather rocks and things like that. But, and I love this little quote here, we are still only beginning to explore the complex relationships between root traits and functions, right? And this is only a quote from a couple of years ago. And one of the reasons for this is possibly due to how roots or fine roots were traditionally studied. So in a traditional sense, what we used to do is we used to go dig up some roots, and we applied what we call a diameter sort of cutoff approach, or this is the traditional approach, where everything under an arbitrary sort of size diameter gets considered as one unit or one functional unit, and we consider that as one sample. Now, the problem with that, as you can see here, is we have all these different numbers. So we have 1, 2, 3, and 4, and these different numbers represent different root orders. And so these are counted by starting from the most distally produced roots, which are the first order, and counting backwards towards the middle of the root system, or the stem of the root system, if you will. And so we have our first, second, and third orders here. Now, with this sort of traditional approach, or this diameter cutoff approach, it made between species comparisons quite difficult. And the reason for this is because of the vast sort of diversity of morphologies of root systems in the world, right, or in the environment. And so here we have two different tree species that I've taken from my study. Up the top here, this is the tulip tree, and down the bottom here, I believe this is maple, but I could be wrong. And so, so if we were to apply this sort of traditional cutoff approach, what that may mean is with the tulip tree up the top here, we may select everything from this sort of branch down. But with the bottom tree species here, we may select the entire thing. So you can see that clearly there's there's a lot of differences in the morphology here. So it doesn't really facilitate these species to species comparisons. Now, one of the sort of newer ways of classifying fine roots is using this functional classification approach, or an order-based approach, where we have these groupings based on what the fine roots actually do in the environment. And so we have a broad grouping here, which are absorptive fine roots. And these are typically the first and second order fine roots. And then we have another broad group, which is the transportive fine roots. And these are typically things that are 4, 5, and higher. And so we have these two broad classifications here. And I mentioned before that they do a whole bunch of different functional roles in the environment. And so what we're looking at here is 6 different measures, or different measures of functionality, or different metrics for different fine roots. And so what we have on the x-axis is we have root order. So we go from first up to 4 and sometimes to 5. And on the y-axis, we just have a different functional measure. And you can see that with increasing root order, so as we go higher number here, we get to more transportive capacity. So with a higher root order, we start to get less uptake capacity, we get less respiration, we get less mycorrhizal colonization. And on the flip side, as we go up towards the transportive fine roots, we get more transportive capacity, hence the name, greater lifespans, and greater CN ratios. So we're seeing these very different functional roles for these fine roots. And traditionally, they would have been classified as one unit together using the 2 millimeter diameter cutoff approach. Now, when we redefine roots in this way, so going from this traditional cutoff approach to more of a functional order-based approach, we get far better estimates of nutrient and carbon cycling in the environment. It also facilitates study-to-study comparison, so we're able to compare different morphologies to each other. And that's all fine and dandy, right? But I'm here. I'm not a root biologist, right? So I think this is cool, whatever. But I'm a microbiologist. So my interest here is how do these different functional roles impact microbiologists? You know, where are these microbial relationships changing because we're seeing different functional roles of these fine roots in the environment? And so before I get too heavy into the microbe side, I just want to have some introduction here. So the plant root microbiome, the microbiome is simply just all of the microbes in or on an organism or in the environment, as well as their combined genetic content. For the plant root microbiome, a lot of these microbial associations are absolutely vital for healthy plant functioning. And so they're very important for plant growth. A lot of these microorganisms bring in nutrients for the plant. And this can be particularly important in agricultural settings, you know, for things like biomass or yield. They're really important for plant health. So a lot of these plants would just not thrive at all or grow very well without these microbial associations. And we'll talk about some of these key microbes in a second. And they're really important for plant defenses. And some of the ways these microbes help is that they just take up like physical space to stop pathogens from colonizing and stopping them from getting in. And a lot of microorganisms like to produce compounds which antagonize or harass other microorganisms like pathogens, and it stops them from also colonizing and causing damage. So a very important system here, and it's a very intricate and I dare say ancient sort of association or like relationships here. So before we go further, I would just want to very briefly mention break down the root microbiome into the different compartments, because it's a very sort of dynamic and complex environment. And so what we're looking at here is a plant root with its tightly attached soil on the outside. So this is just like a cross section of a plant root. And so there's a whole bunch of different compartments. And the reason for this is the plant has different degrees of influence on the microorganisms based on the compartment it associates with. And so for our first compartment, we have the endosphere. This is all of the microbes that exist within the plant root itself. We have the rhizoplane. This is the microorganisms that exist on the surface of the plant root. So you have this yellow sort of outline here. We have the rhizosphere. And the rhizosphere, I write here the Wild West because the definitions of the rhizosphere have been absolutely ludicrous for the last decade, and people just do whatever they feel like, right? But it seems to be we're coming to a bit more of a consensus that the rhizosphere is the tightly attached soil on the root itself. And this typically means you need to wash the soil off the root surface. So it's not like the shaken sort of soil or anything like that. In this schematic, like I was saying, the Wild West, back in the day, the rhizosphere used to be everything that you can see here, but that's not quite true anymore. And lastly, we have the ectorhizosphere, and that is the rhizoplane, which is the surface attached microorganisms, as well as the tightly attached soil, so the rhizosphere. So it's a blending of the rhizoplane and the rhizosphere. Now, in a very simple sense, plants will send carbon down to their roots to encourage these microbial interactions. And this carbon is usually in the form of exudates or other types of rhizodeposits. And what happens is the microbes, they sense this carbon, they go into that environment, and then they can colonize, right? And typically, not always, not all microorganisms are beneficial, but typically these microorganisms then will provide something back in return, and it's typically in the form of nutrients, so things like phosphate or other forms of nitrogen. Now, the best or the most well-known system of this is the mycorrhizal fungal system. And so here we have two broad groups of mycorrhizal fungi. We have the ectomycorrhizal fungi and the arbuscular mycorrhizal fungi. For the ectomycorrhizal fungi, they grow between the cells of the plant root. So it creates this sort of like a net. It's pretty cool to look at under a microscope. And then we have the arbuscular mycorrhizal fungi, which grow within the cells of the plant root. So again, very ancient sort of associations here. And so these fungi associate with the plant root and they grow out into the environment, and that increases the surface area of the plant root because they can bring back water, they can bring back nutrients, and the plant thrives because of that. So what exactly is the problem here, right? The problem is microbiologists like me, we've been using this traditional approach for a very long time, where we just take roots and we homogenize these roots, and then we just say that's our sample, right? And that's our signal that we're measuring. And here, this is really well highlighted by a colleague of mine, Suzanne Fleischmann, who's currently putting together a big review about different methods of root microbial sort of techniques. And she did a big deep dive into the literature. And what we're looking at here is a pie chart. And looking at studies that separated out their roots prior to microbial analyses. And you can see that the vast majority of these studies, 90%, and there's an updated figure which is about 93%, so over 90% of these studies do not separate out their roots prior to microbial analysis. So this is a big problem. Wow, this is a big problem in my field at the moment where we're just homogenizing roots. And so given that I've talked about how these fine roots differ based on a whole bunch of different metrics, we're talking about respiration, morphology, nutrient, water flux, all of these different metrics, they do change by different fine roots. And given that, do we not kind of expect that maybe these would represent unique microenvironments for microbial colonization? And so to explore this, we started off a project and we had a couple of different hypotheses. So our first hypothesis was that fine roots with different functionality, so like our absorptive fine roots and our transportive fine roots, they would house separate or distinct bacterial compositions. And because of the greater sort of metabolic capacity of the absorptive fine roots, we hypothesized that we'd actually see taxa or bacteria that are really well known for like rapid carbon usage. We thought we'd see more of those in that type of environment. We expected that we'd see stronger microbial filtering in that because we're seeing so much flux in this environment that the host might have a stronger selective pressure on the surrounding microorganisms in that environment. And lastly, because again of the greater metabolic flux here, we'd expect that the microbial carrying capacity would be greater. So there'd be greater abundance of bacteria there. And so to explore these hypotheses, we used at the time, a 23-year-old common garden plantation, originally made with 16 different tree species. It was built with a complete randomized block design. So here we have the top of the common garden, the front of the common garden. This is out at the Rock Springs Agricultural Farm out at Penn State University, and it's run by Dave Eisenstadt at the moment. Here we have some of the sampling team, and this is Terry Bell, my old advisor here. We chose 6 different tree species to get a mixture of coarse and thin rooted tree species, as well as half like 3EM or 3EM and 3AM, so arbuscular and ectomycorrhizal associating fungi. So here we have some nice different pictures of the tree roots here, and you can see the morphologies are quite distinct from each other. And so from these pictures, it's a bit hard to see because the boxes are quite small, but the absorptive fine roots are highlighted in orange here, in these orange boxes. What we call the intermediary fine roots are in green, and intermediary is just, it's on its way to be a transportive fine root, so the morphology changes a bit. And we have the blue one here, which is the transportive fine roots. And you can see that particularly for the tulip tree down the bottom here, the transportive fine root is very distinct. But for other trees like the pine tree or walnut, you can see it's not really, you could very easily homogenize that into a sample. And so we collected these roots. We sorted them into absorptive, which is 1 and 2, intermediary, which is 3, and transportive, which is 4 and 5, fine roots. We also took bulk soil, so we could actually look at how the surrounding microbes were being recruited or the selective pressure being placed on those by the host. We sonicated these roots to release the ectorhizosphere. So in this case, we're looking at the rhizoplane mixed in with the rhizosphere, so the two together. And we also did, for a few of these samples, we did some flow cytometry to look at that idea of microbial carrying capacity, so to quantify abundance. And so this is a compatibility issue, but this should be a nicer color, but it doesn't matter. But anyway, the colors carry through for the rest of the talk. Anything in red is an absorptive fine root. Anything in green is intermediary. Anything in blue is a transportive fine root. In this case, the purple are bulk soil bacteria. And so what we're looking at here, this is an ordination or a constrained analysis of principal coordinates. And each dot is a bacterial composition or a microbiome, if you want to conceptualize it in that way. And these dots are plotted based on how similar or dissimilar they are to another sample. Right? So this gives us an idea of how similar or dissimilar these bacterial compositions are from each other. And what we can see is that we get nice clustering of bacterial compositions or microbiome samples, if you will, based on different fine root functionalities. So they're separating out based on the fine roots they associate with. And the bulk soil is also different to the root associated microbes. And we saw this pattern for all of the tree species that we selected as well. And so we're seeing differences according to root order for all of these tree species. Now, one thing that we notice is that when you look at these ordinations, you can see these the purple ones here, the bulk soil ones, there's a general sort of pattern where they appear to sort of cluster a little bit closer to the transportive fine roots. So the blue ones here, it's probably the most obvious for maybe this one down here, which is oak. And so we wanted to look into that a little bit deeper to see how that's impacting. So because this sort of indicates that the plant's having less of a selective pressure on those microorganisms. So we looked into this a little bit more. And what we're looking at here, these are Bray-Curtis dissimilarities relative to bulk soil. And what that means is I took the bacteria from a root and I took the Bray-Curtis values relative to soil so I can see how similar they are to each other. And the way this plot works is anything closer to 0 on the y-axis means that it's more similar to bulk soil. Okay, so it's actually the opposite in your brain. You think it'd be higher, but no, it's the opposite. Okay, so anything higher up means it's more dissimilar. And so what we see here is that for absorptive and intermediary fine roots, they have bacteria that are more dissimilar to the surrounding bulk soil, whereas the transportive fine roots here, their bacteria are more similar to the surrounding bulk soil. So this is indicating to us that there is less selective pressure being applied on transportive fine roots on the surrounding sort of bacteria in the environment or in the soil. So what about the taxa? Who exactly are driving these patterns between different root orders? And so here we're looking at different phyla, which is just a taxonomic rank. These are all for bacteria. And so there's a whole bunch of different taxa here. Most of them don't mean anything, but the ones that I really want to point out are these three here: the beta proteobacteria, the acidobacteria, and the Bacteroidetes, right? Now, the way this plot works is this is relative abundance delta change, and this is relative to absorptive fine roots. So what that means is anything that is in the negative direction means there's more of it in absorptive fine roots, and anything in the positive direction means it's more in that specific fine root. So in this case, the green is the intermediary and the blue is the transportive. Does that kind of make sense to you guys? So anything negative means more in absorptive, anything positive means more in that specific root order. And so what we see here is that the beta proteobacteria and the Bacteroidetes, there's more of those in the absorptive fine roots. Now that's a really cool pattern because a long time ago, back in 2007, an awesome microbial ecologist by Noah Fiera set out to figure out some general sort of functional roles of bacteria in the environment. And what he found was that the beta proteobacteria and the Bacteroidetes correlate really positively well with carbon mineralization. So these taxa are really good at using carbon and breaking it down really quickly. And in the exact same paper, this is perfect for my study actually, what he found was that the acidobacteria had the opposite trend. So it had a negative relationship with carbon mineralization. And so what we're seeing in this study here is a perfect parallel of our data with the previous literature, where we see more taxa that are known for carbon mineralization or rapid carbon usage as being more abundant or relatively abundant in the absorptive fine roots. And this makes sense because absorptive fine roots, metabolic flux, so we should expect to see more of those taxa there. So this sort of tells us that we can start to separate out the taxa into, at least it sort of indicates that copiotrophic bacteria, so things that like to use lots of different carbon or easy to use carbon resources and they grow really quick, we see them more preferentially colonizing the absorptive fine roots relative to the transportive fine roots. So that's cool. So we're seeing differences in the taxa, we see differences in composition. How about the carrying capacity, the physical abundance? And so this is flow cytometry data. On the y-axis, we're looking at cells per gram of dry root. And on the x-axis, we have our different fine roots. So we have our absorptive, intermediary, and transportive. We did this on 3 different tree species. And what we found is that for 2 of the 3 tree species, so we have walnut here and pine tree, we saw greater bacterial abundance in the absorptive fine roots relative to our other fine roots. So it's particularly obvious here in the pine tree. We're seeing way more bacteria in our absorptive fine roots. This again makes sense. Absorptive fine roots, metabolic flux, should be more carbon there, right? We didn't see this for the tulip tree. This could be because the tulip tree is such an ancient species that their roots are really, really funny. They're like little clubs, so maybe something funny is going on there, but the error bars are quite large, so it could also just have been, just didn't work out. But at least the general trend we see is that there are more bacterial abundance in absorptive fine roots. I mentioned that we had picked half AM and half EM associating trees. And so I'm just going to very briefly talk about that pattern too, because people love mycorrhizal fungi. And so for each of our fine roots, so absorptive, intermediary, and transportive, we do see distinct clustering for AM and EM associating trees. So this is a broad pattern that we're seeing as well. The black dots here are arbuscular associating trees, and the triangle purple ones here are ectomycorrhizal associating trees. And so this is where I plug a good friend of mine, Kaylin Yates, who published a lot more on this sort of pattern about the mycorrhizal fungi and how they're influencing the tree properties. So if you're interested in more of that mycorrhizal sort of influencing soil, highly recommend going and reading his paper there. In terms of the taxa driving the patterns between the two, the one taxa that stood out the most was acidobacteria, and it was overrepresented in the ectomycorrhizal trees. And again, there's perfect literature a couple of years back that showed that arbuscular mycorrhizal fungi are known to antagonize acidobacteria a little bit. And so the fact that we're seeing more of them here in our EM associating trees makes sense. So it's, again, perfect parallel to the literature here. So just to summarize the first project here, we see differences in bacterial composition according to fine root order. A lot of the taxa that drive these differences are copiotrophic in nature, so things that like to break down carbon really easily or grow really quickly. We saw differences in microbial filtering, where the absorptive fine roots applied a greater selective pressure on the surrounding bacteria in that environment. And we also saw differences in microbial carrying capacity, where we had greater bacterial abundance in our absorptive fine roots. So after finishing this project, we're like, well, you know, just like any science project you finish, there's a billion questions that you need to answer, right? And so we published this, it got a lot of attention, we're super happy with that. But we had a whole bunch of questions still remaining. So the first one was, in that first study, we studied the ectorhizosphere, which is a mixture of the rhizoplane and the rhizosphere, right? So the question here is, which compartment is actually driving that pattern? Is it the rhizosphere or is it the rhizoplane, or even both? We just don't know, right? Because at the time we chose to study both at the same time. Secondly, are the microorganisms associating to different fine roots because of the resources available at each fine root? Is it just simply that there's different carbon resources, so they're being pulled into that environment? So we wanted to have a look at the metabolomics of this system as well. And lastly, are the microbial functions different? You can get differences in composition, but you may not get differences in microbial functionality in the environment. So we were interested in looking in that as well. And so after finishing the first project, I wrote a little grant for one of the national labs, Pacific Northwest National Laboratory, and we managed to get a bit of funding to do metabolomics, and we work with them to do this second project here. So this is our follow-up study. For this study, we chose 4 different tree species, 2 AM and 2 EM, and we had 2 coarse sort of rooted ones and 2 thin rooted sort of species. For this study, we collected absorptive and transportive fine roots, and we took the rhizosphere and the rhizoplane from each one. Now for the rhizosphere, we did amplicon sequencing. Just very briefly, amplicon sequencing is where we use a target gene, such as 16S rRNA, so the ribosomal gene, or ITS, the ITS region on one of their ribosomal genes. And it allows us to do high throughput sequencing to figure out which bacteria is carrying that specific copy of this gene. So we did that for the rhizosphere. For the rhizoplane, we did a little bit more work where we also did amplicon sequencing, but in addition to that, we did metagenomics. And metagenomics is where you sequence entire genomes from the environment, so you can get their functional roles as well. And we also perform metabolomics, so we could look at the resources that were available at those sort of microenvironments as well. And so our hypotheses for this project was, given that we saw the differences in the ectorhizosphere in the first study, we hypothesized that actually the rhizoplane would probably be driving most of these differences. So we thought that would be the most important compartment, and we would see these differences both in composition as well as microbial function. We also hypothesized we would see a general trend where the rhizoplane and the rhizosphere would be different from each other at both of the different root orders, so our absorptive and our transportive fine roots. And we expected that because of the sort of metabolic flux that goes on in the absorptive fine roots, we thought we'd see a lot more like simple sugars, things that are very easy to use, whereas we'd see maybe some more defensive-based compounds in the transportive fine roots because it's trying to prevent things from, trying to get in and be pathogenic. And so the first pattern, what we're comparing here is I'm comparing the rhizoplane to the rhizosphere in each specific root order. And so what we saw was that for the absorptive fine roots, we saw significant differences between the rhizoplane and the rhizosphere, but we did not see that for the transportive fine roots. So the differences between the root compartments were only seen for the absorptive fine roots, not transportive fine roots. So for this plot here, this is just another ordination. For the different shapes, so the squares here are the rhizoplane and the triangles are the rhizosphere, and the different colors are just different tree species. And so I really like this graph because it looks like a sort of a mirror copy of each other because they're different compartments. So it's sort of like being like pushed up a little bit, and that's just different degrees of host influence there. So we're seeing different microbes. What about the taxa? So some of the taxa that appeared to differ between the rhizosphere and the rhizoplane, one of them was the gamma proteobacteria. Now, with the recent changes in microbial taxonomy, which I can talk to you for a week because no one was really happy with it, but the beta proteobacteria that I was talking about before, the one that's very copiotrophic, likes to use those very easy to use resources, they were moved into the gamma proteobacteria. And in fact, the gamma proteobacteria is majority made up of the beta proteobacteria. So the shifts that we're seeing here are primarily driven by the beta proteobacteria, where we see more of them in the rhizoplane, so on the surface of the root, as opposed to the rhizosphere. On the other side, or on the flip side, there's another sort of class here, which is the Verrucki microbia. We see more of this class in the rhizosphere as opposed to the rhizoplane. And a lot of the microorganisms in this class like to associate with other organisms, so like unicellular sort of eukaryotes and things like that. So it makes me think that maybe there's more of these organisms in the rhizosphere, which are just carrying these microbes as well, right? Because it's also a hotspot of activity, because we do get a lot of, things going on there. So you'd have predators as well and things like that. For this comparison, I was comparing the rhizoplane, so the same root compartment, so the rhizoplane, between the absorptive and transportive fine roots, or I was comparing the rhizosphere between the absorptive and transportive fine roots. So it's the same root compartment, just comparing the root orders, if that makes sense. Yep. And what we found was that the rhizoplane was significantly different between the root orders, but the rhizosphere wasn't. So we're seeing more of a host influence on the rhizoplane than we are on the rhizosphere. There seems to be a general influence on the rhizosphere, but there's more of a spatial pattern for the rhizoplane. And same thing here, same sort of ordination that we were looking at before. In this case, the different shapes are different root orders, with the circles here being the absorptive fine roots and the triangles being the transportive fine roots. So we're seeing differences with different functional roles for the rhizoplane, but not the rhizosphere. And what about, again, the taxa? Who's driving these patterns? And again, our favorite one, gamma proteobacteria, which is beta proteobacteria, really at least in this study. We see way more of that in the absorptive fine roots as opposed to the transportive fine roots for the rhizoplane. And again, this mirrors the pattern we saw in our first study, because absorptive fine roots, metabolic hotspot, we'd expect to see more of them in that environment. For the root metabolome, so the resources available here, this is again another ordination. In this case, the shapes are different root orders. We saw significant differences in the root metabolomes for the absorptive and transportive fine roots. So the resources available at these different fine roots are different from each other. A lot of these differences were primarily driven by sugars, which is one of our hypotheses. So things like sucrose, tagatose down here, fructose, things like that. So the flux between those was driving a lot of the differences between these different root orders. And we also saw some defense compounds, such as catechin, which is, I think it's that one there, maybe, as being more of that in the transportive fine roots. So again, our hypothesis was on point, where we were thinking there was more sort of defensive sort of compounds in the transportive fine roots. And lastly, I had talked about microbial functioning. You can get differences in composition, but the functions may not necessarily differ from each other. Luckily, we did see differences in functioning, which is fantastic because it's a lot of money to do metagenomics. And so for the absorptive fine roots, what we found was that we saw an overrepresentation of functions functions responsible for sugar transport systems. So we're seeing a lot more sort of movement of sugar in that type of environment, which makes sense. There's a lot more sugars in that environment, as we saw with our metabolomics data. And we also saw a greater overrepresentation or greater abundance of isopeptidases and endopeptidases. On the flip side, in the transporter fine routes, we saw overrepresentation of dipeptidases. So I'm not entirely sure why we're seeing this pattern here. Probably has something to do with maybe amino acids or maybe the energy you can pull from trying to break those things down. And urea transport, which is completely random, and I have no idea why that's there. If anyone knows anything about urea in plant roots, definitely let me know. Just to summarize those patterns. So we see differences between root compartments for absorptive fine roots, not transportive fine roots. We saw differences between root orders for the rhizoplane, which is the microbes on the surface of the plant roots, but not the rhizosphere. And these differences we saw with the rhizoplane were mirrored by composition and functional data as well. So we see differences in both. And we also detected differences in the root metabolome between the different root orders, so absorptive and transportive fine roots. And this was largely driven by differences in sugars and, in this case, a defensive compound, which is catechin. And so what about the implications? You know, this is all fine and dandy. Who cares, right? What is this actually? What's the importance of this data? So the first one is that really each root order, or at least each functional role, they are unique microenvironments for microbial colonization. And so the sampling methods that you use to study these root microbial associations are quite important. I've shown you that the composition and even the functions can change between different root orders or groups of root orders. And so this really highlights how if you homogenize your roots, you can really mask those signals that you're trying to detect. And these days, a lot of people are trying to do what we call genome-wide association studies, where they look at individual SNPs, which is just a change in a nucleotide, to sort of link that to specific bacteria or specific taxa. Now, the problem with that is those could be quite subtle signals. And so if you are homogenizing your roots or not really separating them out properly, you could be masking those very subtle signals. So you could be hiding the things you're trying to detect. And I love leaving this talk with this quote here. This is from Luke McCormack's paper in 2015, New Phytologist. And he said, compared with their closest above ground analogs, functional divergence between absorptive and transportive fine roots would be equivalent to that observed between leaves and twigs. And if I was studying the leaf microbiome, I wouldn't have a twig in my leaf microbiome. And so for the roots, we should definitely be separating them out as well to really discover or uncover these types of relationships or what the roots are actually doing in the environment. And so before I finish off my talk, I just wanted to just spend a minute or two about some of this sort of future work that we're doing here, which is very much trying to figure out or decipher some of these microbial relationships with plants. And so some of the work we're doing here is looking at developing what we call synthetic communities of microorganisms. So by putting in microorganisms that we know in with a plant, we can actually begin to figure out who's interacting with who, to look at the feedback and to understand what's important, what's the host microbial feedback in the system, what's driving it. And so here, I love this picture. This is a 96 well plate, and in each well of this plate, except for 3 of these where they didn't grow, there is a unique bacteria in each one. And so with this lighting, it might be a bit hard to tell, but you can start to see different colors. So that bacteria is a little bit different to this one here, and that one's a bit darker. So there's all these different phenotypes there. So we're capturing a big diversity of microorganisms with this. In this case, it's a high throughput culturing system. We're also developing these techniques in the lab. This is called a flow pot system, where we can actually sterilize the entire unit itself. So that way we don't get microbial contamination coming through. And so this is just really a glorified syringe. It is a syringe, just a bit more glorified. And so the idea of this is the entire unit can be sterilized. We can sterilize the substrate. We can grow plants in this, and then through the bottom or even the top if you want, but the bottom's a bit easier, you can push in your own microorganisms and start to manipulate the system to figure out what's going on. Lastly as well, so many of you may know that we can only culture maybe 1% of all microbial diversity in the environment. There's a whole bunch of things that you just physically can't grow, or we just can't do it with our current technology. And so we are also running these experiments where we do these whole community manipulations, where we'll put, in this case, soils under different abiotic stresses to shift a whole community towards a certain direction. or at least adapted to certain conditions, and then we can give those whole communities or those soils to plants to see how those plants then respond under, say, drought stress or under abiotic stress. So it's a whole community sort of inoculum approach as opposed to individual microbes here. And lastly, just want to plug some students in the lab. This is Gillian Gomer and Jacob Scharfeder. And so our lab is really starting to try to break down these relationships between hydrology, exudation, so host feedback, and microbial sort of interactions. And so Taryn's assembled a fantastic team actually to start to really uncover or decipher these relationships to figure out what's going on and what's going on in this sort of fine scale system here. I'd love to acknowledge an absolute massive team of people that have helped me over the years with these projects, as well as a whole bunch of people from Penn State with particular emphasis on Michaela Centenari, Dave Eisenstadt, and Terry Bell, my last postdoc advisor, as well as a bunch of people from Cornell that was super influential in helping me sort of interpret some of this data and sort of like figure out what's going on, as well as the team from the Pacific Northwest National Laboratory who performed all the metabolomics and gave me my grant to do this, as well as the USDA for their funding, the US Department of Energy, and NSF as well. Cool, thank you.
Any questions?
Yeah.
So I've become familiar with research that emphasizing the role of root hairs in the as an important interface between the root and soil microbes. So have you looked at the root hair morphology of these different? Okay.
Definitely, yeah. I think you're right. Root hairs are very, very important. In these studies, we didn't, just because there's an extra step, and this is we're already trying to move the field towards separating out fine roots just in general. I definitely think root hairs have their own unique sort of interactions with microorganisms, as you're suggesting. We didn't look at that into here, but it's definitely an avenue of exploration. So, and I think a lot of people are looking at root hairs. It's one of the big things, at least when I think of microbial studies, people will look at the tips of roots or root hairs, and they'll do a lot of like microscopy work or chemotaxis work. But I've not seen sort of bigger trends where we look at general spatial patterns on root hairs, because that could be quite interesting as well. But yeah, super important.
Anybody from Geneva have any questions?
Cool. Looks like I'm being let off easy. Sure, go for it.
Thorough.
You mentioned transfer root, right?
What was that?
Transfer.
Molharia?
Urea.
Oh, urea. Yeah.
So I was wondering if something that's like, or or didn't select for microbes that use urease as part of their metabolism, and if that could be why you're having accumulation of urea.
100%. Yep. So my understanding is that the use of urea in microorganisms is quite rare. I could be wrong, but there's only a couple of things that I know of off the top of my head that do this type of either like bring it in or metabolize it in some way. It's probably linked maybe to do with nitrogen cycling in some way. I know that with root systems, you can get differences in nitrification and denitrification at very fine scales. So typically roots will have less nitrifiers and more denitrifiers. So I'm wondering if it's more related to that type of system, but it's a complete guess in the dark at the moment. I know, for example, it's a shame I don't have the video, but I know arbuscular mycorrhizal fungi, when they associate with plants, they will specifically bring in denitrifying bacteria to the root surface. And so I'm wondering if it all links up into that system somehow, but it's a complete guess. I have no idea why urea is more of it's there. Couldn't tell you.
This has been a production of Cornell University, on the web at cornell.edu.